It's my pleasure to be here to discuss with you the concept paper on pharmacogenomics in pharmacovigilance. Why is there a need for a new guideline on pharmacogenomics in pharmacovigilance? Well, we all agree that there are large variability in drug response that can be related to genomic variations. So subset of patients may have a different benefit-risk profile. But at the time of marketing authorization, the available information may be limited for genomic subpopulations, although the benefit-risk evaluation could be made for general population. The rare serious reactions may be identified late in drug development, and sometimes only after drug is marketed when the exposure is increasingly large. Currently, the available pharmacogenomic-related guidelines is mainly on drug development in the early phase. So it was considered there is a need for guidance paper on pharmacogenomics in pharmacovigilance to focus on the conduct of pharmacovigilance for medicinal products with genomic associations. The following 5 areas could be the focus for the concept paper for discussion. I will come back to each of these 5 areas. But before I go into the details on the concept paper, I would like to discuss a few general topics, general aspects on pharmacogenomics in pharmacovigilance. This concept paper has been released for public consultation in the beginning of this year, and we have received positive feedback from a number of stakeholders. So the guideline is under development regarding this concept paper. What's the question for pharmacogenomics in pharmacovigilance? It's about genomic biomarkers, about 2 kinds of biomarkers, one for patient selection and one for dose selection. For safety genomic biomarkers, I think you could say generally there are 2 kinds. One is the biomarkers for testing an individual to define risk status for adverse drug reactions. For example, for the idiosyncratic reactions, immune-related, HLA-associated severe adverse reactions. Here we have abacavir-induced hypersensitivity that's strongly associated with the HLA-B5701 allele. We know, familiar with this. And same allele, HLA-B5701, is related to another drug with another adverse reaction. In this case, it's flucloxacillin and liver toxicity. And then we have carbamazepine example. The biomarker HLA-B1502 allele is strongly associated with the Stevens-Johnson reaction in Chinese and the Thai population. But another biomarker, the A3101 allele, is associated in other population than Chinese and Thai population. The 3101 allele is associated with not only Stevens-Johnson, but with less severe cutaneous reactions as well in European Caucasians and in Japanese. And then we have phenytoin-induced Stevens-Johnson, and that is the same allele as for carbamazepine for Chinese and Thai. But for allopurinol-associated Stevens-Johnson, it's a different biomarker. It's the B5801. So what these tell us? For same adverse reactions induced by different drugs, there could be different biomarkers and in different ethnic populations. But the same genomic biomarker could be important for structurally related products like carbamazepine, oxcarbazepine, and phenytoin. The second kind of safety biomarker is to test the individual to define rate of drug metabolism. Here we have mentioned a number of examples for the cytochrome P450 polymorphisms. For 2D6, we have a codeine, tamoxifen. For 2C19, we have a clopidogrel. For 2C9, we have a celecoxib, warfarin, and so on. There are also biomarkers for disease diagnostics and prognosis. We have mentioned quite a lot in the morning, and this is very important in the oncology area. These are usually for safety biomarkers, but lack of efficacy is indeed a safety concern, particularly when the indication is for serious disease. If we look at labeling in Europe, there are three kinds of labeling. One is the testing is mandatory, should be used in clinical use, or recommendation. The biomarker should be used whenever possible or should be considered. And sometimes just for information when the current data is insufficient to make a recommendation. The only example where the genomic biomarker is included in section 4.1, the indication is abacavir. It says before initiating treatment with abacavir, screening for carriage of 5701 allele should be performed. The data supporting this recommendation come from a prospective confirmatory study with a clear clinical validity and utility with high negative as well as positive predictor value. We have the example of carbamazepine. As I mentioned, it's complicated with involvement of two different kinds of biomarkers in a number of different ethnic groups with a different level of recommendation. For the 1502 allele in Chinese, the recommendation is that to test whenever possible to prevent carbamazepine-induced Stevens-Johnson. But for the 3101 allele, the recommendation is only valid for European Caucasian and the Japanese. It says currently there is insufficient data to support a recommendation for screening. But if the patient is known positive for this allele, we should consider the benefit risk. So when we look at the biomarkers that test risk status for idiosyncratic reactions, what do we consider? First, we should identify the clinical variables. Is it about a serious Stevens-Johnson TEN reaction, or is it about less severe reactions? Of course, this may have implication on the genomic biomarkers and the performance of the biomarkers in different ethnic populations. We need to identify the genetic variants. How many biomarkers, which ones, and in which ethnic population. Regarding risk increase, we need to present both relative risk as well as in absolute terms. Regarding performance of a genomic biomarker, we need to know the sensitivity and specificity. For the poor, I'll come back to that, as well as for predictability. What's the positive predictor value and negative predictor value? The negative predictor value is quite important for the safety biomarkers. We need to consider data sources and level of certainty or uncertainty on evidence. And we should consider if the therapeutic alternatives is available, since this is about patient selection. Now the biomarker on exposure. We have a clopidogrel as example for 2C19. It says in pathology section of the product information that 2C19 poor metabolizer status is associated with a diminished response to clopidogrel, but the optimal dose regimen for poor metabolizers has yet to be determined. And then in the warning precaution session, it says based on the literature, patients with reduced 2C19 function have lower active metabolite and diminished antiplatelet responses and higher cardiovascular events following myocardial infarction. There are also labeling regarding interactions worn for inhibitors and a lot of text on the PPI. The pharmacogenetic testing is included in section 5.2, that is the pharmacokinetic section. As for information, it says the testing can identify genotypes associated with variability of a 2C19 activity, but did not say anything about the predictability. And it says there might be genetic variants of other P450 enzymes, so this probably have affect the predictability of this biomarker. We have another example we call Tamoxifen Controversy, because there are a number of studies showing inconsistent outcome in the studies. There are studies showing association between 2D6 genotype or inhibitor use with breast cancer outcome. But there are a number of studies have not shown this association. We could discuss the doses used in the studies, compliance of the patients, inhibitor use, allele analyzed, etc. that could have been important for the confounding in the studies, as well as we don't know if there are other metabolic pathways that can be important. The prospective studies have shown also different studies, different results. Even though there is a strong association between the genotype and drug exposure, it has not been shown there is a predictability for the outcome of tamoxifen treatment. It seems this controversy is unlikely to be resolved through retrospective analysis. The current product information in Europe states the following. 2D6 poor metabolizers have a lower plasma level of the intoxifin. That's one of the most important active metabolites of tamoxifen. Potent inhibitors of 2D6 should, whenever possible, be avoided during tamoxifen treatment. The poor metabolizer status may be associated with reduced response, but the consequences have not been fully elucidated. The available stages have mainly been performed in postmenopausal women because there is no alternative for the premenopausal women. So when we consider the biomarkers related to pharmacokinetics, what do we need to consider? We also need to consider, identify the clinical variables. In this case, is it lack of efficacy or particular toxicity, or is about level of exposure? We need to identify genetic variables. For example, for 2D6, there are a number of alleles, and what are the frequencies in what populations? Regarding risk increase, there is usually gene dose effect. It's not yes or no. And performance of a genomic biomarker, the sensitivity and specificity. Sometimes it's a good association between the genotype method and the poor metabolizers, but sometimes the sensitivity for ultra-rapid metabolizer identification could be very low. We need to know the predictability. Is it to predict exposure or clinical outcome? We need to know the if therapeutic drug monitoring or phenotyping has a place. We need to discuss the data sources and level of certainty on the evidence. And sometimes we need to know the therapeutic alternatives as well, although this is mainly about the dose selection, but sometimes you do need to consider alternative treatment. Now the concept paper or guideline on pharmacogenomics in pharmacovigilance. The first question to address is how to give systematic consideration on the importance of the genomic biomarkers in risk management for safety concerns or lack of efficacy. For new medicinal products, it's generally expected to have data available at approval. But if you have a genomic issue, a genomic association, you should discuss in the risk management plan on the extent of a pharmacogenomic effects and implications of a biomarker in the target population. If the study is done in a particular genotype group, we should discuss whether use in patients with an unknown genotype or different genotype could be a safety concern. If important genomic polymorphisms have been identified but not fully studied, we should discuss this in the RMP. And whether it's considered as a safety concern and to be included in the labeling, it will depend on the possible clinical implications. The second aspect is about when and how post-authorization genomic data may need to be collected. And the effort for the pharmacogenomic evaluation will depend on prior knowledge. If in post-marketing phase, important safety concerns rise, the collection of genomic material may prove essential. One approach could be to encourage collection of genomic samples from every patient who used the drug and developed safety concerns in the initial post-authorization period, and compare the data with patients who do not have safety concerns. Here is a study published in 1982 in the UK. It's about paraxylene and 2D6 activity. There are three groups of patients studied. The first one is neuropathy patients on the drug. The second is without adverse reactions on the drug. And the third one is patients not on the drug. It's a general control. And you see in general population, the poor metabolizer frequency is 8%. But for the neuropathy group, the patients, 50% of the patients are poor metabolizers. And usually more than 60% are extensive metabolizers. But in neuropathy group, it's only 15. So if you look at the dose of paraxylene, even though it's a little bit lower compared with the patient group without adverse reactions, this is still too large for the neuropathy patients. Now the level and type of evidence for signal assessment. This has been addressed extensively in the morning. I will not go into detail, but post-marketing collection of genomic material is important, and the carbamazepine is a good example on that. In general, the CDC, ACC criteria for evaluating genetic tests should be followed. The analytical validity, clinical utility, as well as ethical, legal, and social issues. The next area is about risk minimization in the labeling. And I leave this to the next speaker on the product information and on genomic information in the product information. I just mentioned there are two kinds of risk minimization measures. One is routine, and that is SPC or product labeling. Another one is additional risk minimization measures, and this may include restricted access to the medicinal products based on specific tests or patient registry or additional educational material to patients or prescribers. So these are the SPC guideline regarding genomic information, what and where to put. I think the next speaker will address this in more detail. Last point, but not least, is the question on how to measure the effectiveness of risk minimization measures. This is challenging, as usual, but for this area, it could be more challenging. It could be studied in different levels to study the awareness and the knowledge on the recommendation on the genomic biomarker use. Or you can study are recommendations followed, why or why not. and how to measure the effectiveness on the risk minimization measures when it's mandatory or just for recommendation. If there is a warning for interaction, probably you could study drug utilization pattern. Currently, there is work ongoing to develop good pharmacovigilance practice module. Maybe we can learn from the GVP module on tools and methodologies for this topic. Here's just one example that 1502 allele for carbamazepine, Stephen Johnson. It's a beautifully designed study prospectively. They have identified 1502 carriers and avoid carbamazepine in these patients and show a strong association with a decrease in the incidence of Stephen Johnson with carbamazepine. This is my last slide showing some challenges in this area. The post-marketing genomic data collection could be challenging. The level of certainty on the evidence or uncertainty. The implementation of genomic biomarker use that could be different in different parts of the world, and the measure of effectiveness of the risk minimization activity. I stop here. Thank you for listening.
Thank you very much. Do you have any burning question to the speaker, please?
Thank you very much, Qun-Ying. Just an addition regarding, which I think we can learn a lot from the tamoxifen studies. And there were some very large studies published recently where they used the tumor DNA for genotyping, which is awful since gene deletions occur in the Q22 locus in the tumors. And the data were not in hard environment equilibrium at all, which was, and they were, so to say, negative. They didn't find any association, but the two papers, two big studies are really almost, I think, asked to be retracted now. And if you then skip these studies and go into the most bigger studies in the late years with between 500 and 1400 samples, there seems to be a tendency actually with the hazard ratio about 2 for 2 to 6 poor metabolizers. So I think with the bigger studies we get better information. And please be very careful not to use tumor DNA for any genotyping studies if you're studying the host genome.